what a goal is to solve a circuit. And hopefully what I'll show you today is that we are going beyond what is considered solved. So neurons in our brain are often classified as inhibitory or excitatory, depending on how they affect their postsynaptic partners. However, in a growing number of regions, including the hippocampus, habenula, auditory brainstem, and spinal cord, you have neurons that release both an inhibitory and excitatory neurotransmitter. Now, on the surface, that doesn't make sense. Inhibition and excitation are going to cancel each other. Why push the brake and accelerator at the same time? This is the problem that have puzzled retinal scientists for more than 25, 30 years when they realized these beautiful starburst amacrine cells, which lie in the heart of the direction selective circuit, are both GABAergic and cholinergic neurons. I'm going to try to convince you today that, in fact, by finally controlling the spatial temporal properties of GABAergic and cholinergic transmission, the system can actually offer a way to provide neural circuits with a finely balanced inhibition and excitation. And this is critical for high fidelity computation of direction. But before I go to that, let me summarize 50 years of work that led to the solution of the DS circuit. So the DS circuit direction is coded by the retina and outputted to higher brain regions by these directional selective ganglion cells. These were discovered many years ago when people moved a stimulus across this receptor field and saw that it didn't respond, and the same stimulus caused a robust response when the stimulus moved in the opposite direction. Okay, and that's called its preferred direction, and that's called its null direction. Now, if you move the stimulus in orthogonal directions, you get a mediocre response. Now, there are four types. There are many types of DS ganglion cells in the retina, and what I'll focus on are the ones that code the cardinal directions. Okay, there are four types of these. These are the on-off kinds of DS ganglion cells. Now, the main focus of the last 50 years of research was how does these cells get this, acquire this direction selectivity, and beautiful work from Winfried Denk's lab using 2-photon calcium imaging showed that the starburst amacrine cells played a key role. So when they measured calcium in the distal dendrites and moved stimulus from the soma to the dendrite, they saw a very, very robust calcium signal in the distal dendrites. And this is where the neuron is thought to release acetylcholine and GABA from these distal dendrites. Note, there's no axon to these neurons, so it's a kind of dendritic synapse. Now, if you move the stimulus in the opposite direction, going from the distal tips to the soma, then you get very little response. So the DS cell, these amacrine cells, the dendrites of these cells can themselves compute direction. Now, while these can compute direction, the next challenge is how does that direction get transferred to the ganglion cell and output as spiking to the rest of the brain? Now, this is non-trivial because if you connect both these sides of the dendrite to a ganglion cell, you run into the problem in that it'll be direction as the stimulus moves right across the receptor field. They'll cancel out from both sides. So you need a very, very selective wiring. And indeed, again, Winfried stood to the challenge, invented scanning block electron microscopy, and showed that these cells were actually asymmetrically connected to ganglion cells. So that's what's shown over here. You see these radiating dendrites of starburst pointing in different directions, but they connect to very, very specific populations of ganglion cells coding the cardinal directions. Okay, so in that way, the direction selectivity can be transferred to the next layer. In this case, you have images moving across the retina, stimulating the photoreceptors, which stimulate bipolar cells and stimulate both the on and off layers of these ganglion cells. But importantly, the starbursts are only connected on the null side. So when the stimulus moves in from the null side, they activate the dendrites in the appropriate direction, and that provides a very powerful inhibition and shuts off spiking in the directional selective ganglion cell. On the other hand, if the stimulus comes in from the preferred side, you don't have the starburst, and it's going in the opposite direction that the dendrites are activated in. So you don't get that powerful inhibition, and you get beautiful spiking. So again, this was back almost 10 years ago. This was the model put forth, and the circuit was considered solved. So when we looked at the circuit, the major problem we saw was, and this is kind of a general problem in the brain, we know that feed-forward inhibition is extremely important. And if you're going to use different neurons to do that, you're going to run into the basic problem of how do you set the gains of these synapses to be balanced under diverse conditions. So in any part of the brain, when you need inhibition and excitation to be balanced, how does that occur when you're using neurons with very distinct biophysical properties? For this to happen, you need the gains to be set in a very, very set manner. And to give you a better appreciation of this, let's consider contrast, for example. Okay, so when you have the circuit functioning under high contrast, the fidelity of transmission is very high, as you see over here from the trial-to-trial variability. Okay, this is spiking in a DS ganglion cell. However, when you lower the contrast, what you see, the fidelity of transmission is greatly decreased. So now what you see is the excitation, the spiking response bouncing a lot in time. So if excitation to the ganglion cell is going to fluctuate in time, and you need inhibition to clamp that down, how do you do that? Because your inhibition and excitation are not going to be balanced anymore. Okay, in this simple circuit. And to solve this, we hypothesized that the retina uses two tricks. Sorry. Showing that even when you use a high contrast, you get a beautiful direction selectivity. And this is plotting the response against different directions and showing that it likes this direction. If you lower the contrast, even though you have very few spikes, it's beautifully directionally tuned. Okay, again saying, even though you have these fluctuations, the balance of inhibition and excitation is maintained. Okay, you always have more inhibition than excitation in the null direction, and more excitation than inhibition in the preferred direction. Okay, and again, the main question is, how do you balance this inhibition and excitation over diverse conditions? And the tricks the retina uses. So what we're going to try and describe is a slightly different configuration of the circuit. Okay, so instead of having feed-forward excitation and feed-forward inhibition, what we're going to propose is that, in fact, it's acetylcholine and GABA that's driving the system. The bipolar cells activate the starburst, but not the ganglion cell directly. Okay, so this is a fundamentally different plan of the retina, so to speak, where normally you have excitation going through bipolar cells being modulated by inhibition. Here I'm saying, no, the excitation is not reaching ganglion cells directly, but the bipolar cells are stimulating acetylcholine and GABA from this network of starburst ganglion cells, starburst amacrine cells. In the second part, so in the first part of the talk, I'll show you a lot of biophysical details to show you that it is acetylcholine and not glutamate that drives the DS ganglion cell. And I'll show you evidence to show how these can be differentially transmitted to the ganglion cell, and the GABA and the ACH don't simply cancel each other off. Okay, the balance is maintained in a directional selective manner. And then finally, in the second part, I will show you what happens to that bipolar cell input, okay? And how its differential expression of AMPA and NMDA receptors can result in the rerouting of that information through the SAC network instead of directly through the ganglion cell. And this entails novel NMDA-ACH interactions. Preparation we're using is a whole mount retina, where we lay the retina of a mouse on a flat mount configuration. And each circle is a ganglion cell. And with two-photon microscopy, you can identify the directional selective ganglion cells and come down with a patch electrode and move stimuli over and record action potentials. In this case, we decided to play a movie, which was taken from a camera mounted on a mouse's head as it moved its cage. and you see the luminance fluctuations. And this ganglion cell is responding to very specific parts of the movie where the stimulus moves, a stimulus mover feature moves in its preferred direction. Okay. The most striking part over here is when we put on an acetylcholine receptor blocker. Okay, we see that most of the response disappears. Okay, and this was really surprising because, again, it's saying that the response is being mediated by acetylcholine, basically. Now, people had done this pharmacology for many, many years. Since 30, 40 years, they've been putting on ACH blockers. What was different about the conditions over here? One possibility was in the natural movie, the contrast is much, much lower than what the are used in previous experiments, which are just simple spots moving on a high contrast background. So we decided to test the effects of contrast on the role of acetylcholine on responses mediated by different contrasts. So here you see a response mediated by high contrast bars, and if you put on acetylcholine blocker, you lose much of the response, but the remaining response is very robust and it's still directional selective. And this is what people had reported before. However, if you lower the contrast, what you see is something quite striking, is that the response, again, is completely flattened out. Okay, so again, this effect that we're seeing appears to be contrast dependent. You can do this in a little more quantitative way using voltage clamp techniques, but essentially you see the same thing. So again, we're looking at EPSCs now, and you see an on-off component coming as the stimulus edge comes in and out of the receptor field, and that's driven by on and off bipolar cells. But as you increase the contrast, the response gets bigger. If you block acetylcholine receptors, you lose all the low contrast responses over here. And that's kind of summarized over here. So that's the total response. If you block acetylcholine receptors, you see the curve shifts to the right. And the initial component is, if you do a subtraction, you realize that this whole initial component is mediated by acetylcholine receptors. Now, this is a big statement to make because it's challenging the fundamental plan of the retina. And of course, it's just using pharmacology. And the simple assumption here is that acetylcholine blockers are only working postsynaptically. Now, of course, anyone who's done pharmacology on a network realizes there are probably many ways to explain this sort of data. Okay, and we see the expression of acetylcholine receptors in amacrine cells and bipolar cells and other circuit elements. Okay, so how do we realize that this is actually due to postsynaptic acetylcholine receptors? For this, we kind of devised a novel method in which we take advantage of the biophysical properties of the different receptor types and use a sort of deconvolution analysis, which I'll explain in a second. So to understand what the biophysical properties of acetylcholine receptors on BS ganglion cells are, we use the mouse line in which cholinergic was expressed only in starburst amacrine cells. Okay, and then we blocked all other receptors in the circuit and stimulated with blue light. And if we held the cell at different voltages, what we see is that the current decreases as we depolarize the cell. But then when we further depolarize the cell, the current rectifies. And that's kind of summarized in this IV plot. So you see that this acetylcholine receptor has a large inward rectification. You don't get much of an outward current until you go much higher in potential. So it has a distinct signature. And this is kind of consistent with acetylcholine. But it gives us a handle on how to extract this conductance from the whole network activity. In contrast, if you look at the biophysical properties of GABA, you see it's almost linear and reversed around -60, and AMPA receptors are completely linear and reversed around 0. So now we can, given this information and understanding of the biophysical properties of these different receptor types, we can ask, what is the natural response made up of? Okay, and that's 5 minutes. Oh, I really need to rush. So in these experiments, we've also blocked NMDA receptors by putting an antagonist because these really complicate the analysis. And NMDA receptors are always secondary, so we didn't worry about that too much. Now what we did is not put any blockers for acetylcholine or GABA, and we measured the light response at different holding potentials. Okay, and asked, okay, how can we, what are these responses made up of? To understand that better, if you plot the IV of the initial response, you see the response is kind of rectifying. Okay, so if you fit that, you can actually fit it sum of these basis functions and ask what is the weight of each of one of these functions. And when you do that, you realize that the initial response is made up of 90% of acetylcholine receptors. You can see it's inwardly rectifying. Okay, so this is giving us a direct readout of what is being released in the circuit without the use of pharmacology. Okay, so it's a pretty deep technique and it requires a little careful voltage clamp and control of that, but I'll show you that this is possible. So you can do that for different phases, and you see if you go even to the later part of the response now looks a little bit more linear, but its reversal is shifting towards minus 60. And when you try and do that again, fit the curve with different basis functions, you see that most of it is GABA and acetylcholine, and AMPA is minimal. Okay. Now, these are quite subtle changes, right? I mean, is this linear or is this, and how robust is this data? And I'm going to bore you with more biophysical details because this is kind of critical and it's kind of cool in my mind, and I'm a bit of a geek. So you can see over here, the GABA ACH, if you model it with GABA ACH, it fits beautifully. If you try and model the same curve with AMPA and GABA, okay, it does very poorly. Okay, and even though the data, you can see where it's going off. If you go to the high contrast conditions where you do have AMPA, then the ACH and GABA, you still get large errors, and the AMPA and GABA, you get big errors. But now you need all three components to minimize and get a good fit of the data. You can take this a step further when you take the same condition and block acetylcholine receptors. Now in those conditions, okay, the ACH and GABA don't fit the data very well, but the AMPA and GABA fit the data really well. Okay, so these are all we can go through. I'm going to skip these details because the main question is now, given that we can start to pull out all these different receptors in an intact circuit, how does the conductance evolve over time? Okay, and so we did that similar analysis for every millisecond along the light, along the response as the light bar sweeps over the receptive field. Okay, and this is just showing the on response. So when we do that, we come up with this picture. So first we measure spiking, and what we see is that the GABA conductance in the preferred direction is small and delayed compared to this huge GABA inhibition in the null direction. If you look at the acetylcholine, okay, it's equal in the null and preferred direction, and the AMPA is very little. So if you sum the acetylcholine and GABA, you immediately realize there's a window in which you can get beautiful spiking, whereas in the null direction, the null inhibition completely cancels out. It's very powerful compared to the acetylcholine, and you get no spiking. So now this is not surprising in that inhibition and excitation are change in balance. What is surprising is that this is not glutamate and acetylcholine. Okay, and that's what's being revealed over here. And keep going back. This summarizes that this holds for many cells, so you always see that excitation is equal in the preferred and null direction and falls along the straight line. If you look at inhibition, it's much larger in the null direction. If you look at the balance, it's always positive in the preferred direction and negative in the null direction. And you look at the timing, it's always E before I. And if you look at in the null direction, they're always synced up. Okay, so again, it's saying you have a system, and in the endogenous system, when you move low contrast stimuli across the retina, it's acetylcholine and GABA that are driving the computation. How do you actually prove this? And for this, we resorted went back to optogenetics. So we went back to our channel rhodopsin mice and said, okay, let's compare the direction selectivity under normal conditions to that driven by channel rhodopsin. So to do that, we measured direction selectivity in a ganglion cell using really low light levels. Okay, and this is way too low to stimulate channel rhodopsin, but the photoreceptors are intact and much more sensitive than channel rhodopsin, so you get beautiful normal responses. Then we put on a cocktail of antagonists to completely block the photoreceptors, and that you can see the responses are completely gone. Then we increased the intensity of the light stimulus, okay, by almost 5 orders of magnitude. Now you see beautiful spiking again, and the most impressive part is if you look at the directional tuning, it's completely matched with that driven by photoreceptors. Cell types, so you can see the direction in control is plotted against the direction in channel rhodopsin, and it lines up along the diagonal. And similarly, the directional index, selectivity index, is actually stronger in channel rhodopsin conditions because there are fewer spikes. This suggests, this kind of proves that the starburst network alone has the capability of driving directional selectivity. More controls to show that, there's no residual photoreceptor activity. If you do some pharmacology, you can see that easily. Block GABA receptors, you see the response becomes asymmetric, and now you have a lot of activity, and if you put on acetylcholine blockers, the response completely disappears. So there's no residual glutamate activity in this condition. More importantly, if you look at the do voltage clamp analysis under these conditions where photoreceptors are blocked, you see the same kind of phenomenon where you have a larger inhibition in the null direction and excitation is equal. Okay, so this is quite a neat system, right? Because you've got the same neurons that are releasing acetylcholine and GABA, but somehow the response, the EI ratio is different in two different directions. Now, exactly how that happens is not 100% clear. If you look at the connectivity, what we said, the scanning block, and you look at the synaptic connections, you see the starbursts are connected only in the null direction. Okay, and that raises the question when the stimulus comes in the preferred direction, how do you get preferred excitation? Okay, so there is a little bit of things that we need to figure out, and there are two possibilities. One is we need to have preferred sacs on the preferred side because that's what happens when the stimulus comes in, you get acetylcholine. Okay, now why their synaptic connections were not observed in the scanning block EM can be for two reasons. First, you could have different synapses for acetylcholine and GABA, and if the acetylcholine ones are symmetric, you would still see the same kind of. But that would require the synapses to be morphologically different, and that's why they were missed in the EM recordings. The second possibility, which is also as likely, is that perhaps acetylcholine is not being transmitted via conventional synaptic mechanisms. So if you look all around the brain, acetylcholine is actually transmitted through paracrine fashion. Okay, so this acetylcholine sensed by the ganglion cells might come from starburst amacrine cells, but not require a defined synaptic connection. Okay, and that's how we think about how the same neurons releasing acetylcholine and GABA can create very distinct responses depending on the direction of the stimulus. This is just a side note. I mean, what I've shown you is that direction selectivity in the ganglion cells does not require bipolar cells, and it's coming from the starburst network. In fact, this was a bit of contention because a new model that just came out from Sebastian Sung's lab predicted that you need different types of bipolar cells to generate the direction selectivity in the dendrites of starburst amacrine cells. And over here, the data is saying you don't, even without these bipolar cells, you can generate a very robust directional selectivity that matches the endogenous one. So this model needs to be revisited. So let me go to part 2 of the talk, which is to understand what happens to that bipolar cell input. You have a common set of bipolar cells, and you need them to be activated. You need the starburst to be activated in order to release acetylcholine. Why is not that glutamate activating the directional selective ganglion cell itself, right? And if you make measurements in a pairwise fashion from a starburst and a direction ganglion cell, you see the following. And this is, again, increasing contrast stimuli. You see that the starbursts start to respond before the ganglion cell. And that's kind of plotted here for a bunch of cells. And you can see there's at least a threefold difference in the contrast sensitivity of the sacs and the DS ganglion cells. Starbursts are responding at a lower contrast, and this allows them to depolarize and release their acetylcholine and therefore stimulate the ganglion cell. But why is the ganglion cell not responding when the starburst is? So, I mean, we assume that they're coming from the same bipolar cells, and the same bipolar cells are connecting to both these cells. But to verify that, we went back to the scanning block EM, where we identify, and this is done with in collaboration with Kevin Brigman, identifies direction selective ganglion cells using calcium imaging, and then goes and scans the whole block. Seeing a reconstruction of a bipolar cell terminal and a starburst, and the starburst is in yellow. And this is just a sense to give you what kind of resolution we have. It's kind of a beautiful system because you can actually realize what precisely is being connected to what. Right? Which types, there are many types of bipolar cells, and are these bipolars really shared between ganglion cells and starburst amacrine cells? This is just showing you one example of a bipolar cell terminal connecting to this beautiful radiating starburst amacrine cell. Now, if you look at the bipolar cell terminal over here and ask what is its partners, okay, you can see very clearly that they connect, they're shared between the starburst and the DS ganglion cell. Okay, so the same bipolar cell terminal does connect to the DS ganglion cell and the sac. So now we're again, if the same bipolar cell is driving both the cells, then why is the sac responding and not the DS ganglion cell? And the solution to this apparent conundrum came when we considered NMDA receptors. In this experiment, what we did was looked at the spontaneous activity with GABA inhibition blocked, and instead of holding the cell at minus 60, we held the cell at plus 50 millivolts. And the starburst we held at minus 60 millivolts to see the AMPA. And under these conditions, suddenly we see why we were missing what we were missing. Okay, so if you look at the spontaneous activity, they're going completely hand in hand. So whenever you have a large depolarized input to the sacs. And if you do a correlation, you see that they're beautifully correlated over a number of cells. Now, if you block NMDA receptors and you look at the residual AMPA receptors, firstly, you notice there's very little. This is kind of weird because normally a glutamate synapse is dominated by AMPA receptors. And over here, the synapses are dominated by NMDA receptors. I have to read the literature more carefully, but I don't think there's a single case. Maybe Angus can tell me whether you have a synapse, not in the immature system, that's in the mature systems that's dominated by NMDA receptors. And more importantly, that the residual AMPA receptors are very weakly correlated, suggesting they might be coming from a different source. Further characterize them again with paired recordings and shine light this time, you see now the SAC EPSCs, the inputs to the SACs, kind of match up with the NMDA receptor responses in DS ganglion cells. So it is the same kind of bipolar cells, but that mismatch was coming because the the ganglion cells had NMDA receptors. Now, importantly, these NMDA receptors alone can't drive activity. Okay, and that's this is just a confirmation. So if you have a robust spiking response and block AMPA, you see the responses are completely gone. If you take the same cell and voltage clamp it and hold it at different potentials, you see these huge currents. If you look at the current voltage relationship, you see this J-shape IV, which is the signature of NMDA receptors. So in this condition, there is a lot of glutamate being released into the cell. NMDA receptors are sensing that glutamate, but they're not driving spiking. So they're completely silent, okay, which is not surprising. However, picture then that's emerging then is if you look at the inputs to the ganglion cells, you have an NMDA component and you have an acetylcholine component, and the AMPA component is actually coming on much later, which suggests that it's not AMPA that's recruiting the NMDA receptors, but it's acetylcholine that's recruiting the NMDA receptors. Directly, we just use pharmacology, where you show that if you look at a response to the half-max contrast, you find that acetylcholine receptors block the response, as I've shown earlier. But even if you block NMDA receptors, you see a large decrease in the response. Okay? The fact that both transmitters are being used suggests there's a highly nonlinear interaction between acetylcholine and NMDA that causes this robust spiking. Test this out further, what we did is the following experiment to kind of verify this model, in which we had both, we imagined that at the lowest contrast that there would be only NMDA receptors, and as we increased the contrast and we got the sacs to threshold, they would release acetylcholine and GABA, and then that acetylcholine would interact with the NMDA. At threshold, you see that the response is beautifully direction selective again. That means there's inhibitions already kicked in over here. At subthreshold regions, there's no response, so we don't know whether inhibition is kicked in or not. The prediction is not, right? But to reveal the NMDA receptors, all we needed to do was depolarize the cell a little bit, right? And then you see indeed that you get inputs from bipolar cells, but they are non-directional. And this is just the classification. So I guess I finished early. Maybe your timer really set me off. Okay, so let me summarize what I'm trying to say over here. So we have a system where we need to look at the detect motion at different contrasts. When at the lowest contrast, we can't get balance. There's no way to do it because the cells, inhibition and excitation can't be tied in together. And there's no biophysical tricks that the brain can use. But what the retina does use is NMDA receptors to kind of get rid of this and not to allow this imbalance to cause spiking and send noisy information to the brain. Okay, so under these conditions, even though there's glutamate being released, okay, there's no response. Only when the starburst reaches threshold contrast and starts to release acetylcholine and GABA do you start to see responses in the ganglion cell. Okay, and this is a beautiful system because now, however strongly you stimulate it, the ACH and GABA are always tied together because they're emerging from the same cell. So the stronger you stimulate it, with the higher the contrast, the more the GABA and the more the ACH. So this system always stays in balance then. Now, although ACH and GABA are being released by the same cell, they're differentially transmitted to the ganglion cell. And that I showed you was because the GABA was asymmetric and the acetylcholine to the ganglion cell was symmetric. Okay, so this is a beautiful trick to get a very, very balanced inhibition and excitation. And finally, the bipolar cell input doesn't go completely to waste. Okay, you activate NMDA receptors very strongly, and this has the added effect of boosting this weak cholinergic input, especially at a low threshold. Again, notice the NMDA cannot interact in the null direction. It can't boost those responses because they're being shunted, and NMDA is very voltage dependent. So it's a very beautiful system of amplifying these preferred direction and really increasing the signal to noise. And finally, all this put together allows us to have contrast invariant DS tuning, so at every contrast, the inhibition and excitation stays balanced, and you get a very robust computation. With that, I'd just like to thank all the people who did the work. Sansa Sethu Ramanujan did the acetylcholine work. Alex and Amanda did a lot of the balance stuff, and I haven't talked about the other work. And finally, the Canadian funding agency, the Canadian Institute of Health Research, the NSERC, the Foundation for Blindness and Brain Canada. So thanks for your attention.